Good morning, ladies and gentlemen, distinguished guests, fellow researchers, and esteemed colleagues. It is my greatest honor to welcome you today to the second edition of Neurocon, an important gathering for the neuroscience, neuropsychology, and neurobiology communities. As we reconvene for another year, our dedication to uncovering the enigmas of the brain remains unwavering. Our inaugural conference set the stage for groundbreaking dialogues and collaborations, and this year we promise an even more enriching experience. We have an exciting two-day program lined up for you, touching on a diverse array of topics that exemplify the dynamic nature of our field. It's an honor to announce that today the renowned Professor Carl Friston will be giving a talk on active inference, a topic that holds great promise for reshaping our understanding of the brain. Furthermore, tomorrow, our panel discussion on potential vaccines for Alzheimer's disease is bound to ignite stimulating discourse on one of the most pressing challenges that we're currently facing. These are just two examples of what we have prepared for you, and beyond these, you can expect deep dives into exciting new directions in neuroscience, neuropsychology, and neurobiology alike. My name is Cristiana Dimulescu, and I will be your host for the day. Thank you once again for joining us, and I hope you're as excited as I am to get started. I would like, first of all, to mention that this conference is also being transmitted live on our YouTube channel. And for those of you who are watching us there, if you have any questions for our speakers, please use the live chat on our website, that is neurocon.eu, for addressing these questions. And with that being said, I would like to introduce our first speaker for the day, Professor Fabrizio Doricchi, Professor of Neuropsychology and Research Methods in Neuroscience and Cognitive Neuroscience at Sapienza University of Rome. Professor Doricchi's work focuses on the brain mechanisms governing attention control in both healthy individuals as well as those with acquired brain lesions. And today he will be talking about the temporo-parietal junctions in the context of free energy approximation and their role in updating predictions. Professor Doricchi, you have the floor.
Okay, good morning, everybody. Thank you so much for inviting me to this exciting event. I'm thrilled in a way to have the chance to speak for the time I'm left with to a relatively huge argument, which is the function of the temporo-parietal junction. I'll try to convey during this talk all the main infos and the main news about the TPJ that we have reached in a recent review paper, so a theoretical paper, quite huge review. So I don't think I'll be able to convey all the infos you can gather from reading the review in a short talk, but I'll try to give you the basic hints, clues, which I think will help us, I'm quite convinced of this, better understanding of the TPJ. So I divided my talk in basically two parts. The first part will be dedicated to describe what the TPJ, where the TPJ is, and introduce you how we reach this more general idea on the function of the TPJ during the course of the years, and the reflections and talks that started from research on attention and then culminated in a more widespread interpretation of TPJ function. So let me just start with my presentation. Please let me know whether it can be clearly seen from. You can see the slides correctly?
We cannot see anything yet.
Oh, sorry. Why? Should I share the screen?
Yes.
How? Where is sorry.
At the bottom of the screen, you have the screen button for sharing the screen.
At the bottom?
Yes. If you hover.
Okay, found.
Perfect.
Okay, now should be fine.
Yes, now we see your presentation.
Yes, and now now it's fine?
Yes, perfect.
So I should hide this probably.
That's okay. I don't think we see your Zoom. We don't see your Zoom part. Just
okay, okay, just the slides. Okay, yes. Perfect. So the title of all is the roles of the temporo-parietal junction. As you see, there is a singular and plural word because this is one of the main problems that has been dealt with when talking about the TPJ. And it doesn't work. Okay. What I'm going to do is basically exemplify, summarize the idea that we have sketched in this recent review. Okay. So let's first talk about the TPJ, the temporal-parietal junction. Okay. You know, the point with the temporal-parietal junction is that is a quite heterogeneous area. So it is difficult. During the years, there have been several essays and tries to correctly define anatomically the TPJ, the border of the TPJ. So I think a quiet consensus has been reached by defining the TPJ through the areas that border the TPJ, the act as border of the TPJ. So the extrastriate cortex, particularly the MT area, was established the inferior border of the TPJ. The anterior border would be set, but would be coincident with the somatosensory cortex and the temporal auditory cortex here, and the posterior cortex, I would say that basically is defined by the interparietal sulcus, which, as you know, divides, which is this one, this sulcus that divides the inferior parietal lobe from the superior parietal lobe. Okay, so right from this slide, you can check what was thought to during, especially based on functional MRI studies, to be a homogeneous area is not at all, in a way, a homogeneous area. This partition has been established recently by Ilgastrom and Graziano based on DTI connectivity and MRI studies of connectivity. As you will see, it reflects also old histological data that show that the TPJ, even from a histological point of view, is a quite heterogeneous area. What is important about the TPJ to remind is that it has a very late ontogenetical development. And if you compare humans with macaques, it is probably one of the most phylogenetically expanded cortical areas in the brain. So if you take these two facts together with the fact that there are important inter-individual variation in the morphology of the TPJ, you can reach the conclusion, and has been done by many authors, that like other association areas in the human brain, the TPJ contributes to high-order cognitive processes. So if you going ahead, these are the histological maps. Let's go a bit back in history by Brodmann, who individuated basically two areas, BR39 and BR40, which corresponds entirely, according to Brodmann's definition, to the angular gyrus in the inferior parietal lobe and the supramarginal gyrus also in the inferior parietal lobe. While Konstantin von Economo, who actually made much more detailed histological maps in his lab between Trieste and Vienna at the end of the 19th, beginning of the 20th century, found much more histological, made a much more subtle differentiation of this area. He counted at least 6 different histological areas. These all data are now matched by post-mortem studies. This is, I think, is a more recent study by the group of Binkowski. I think the first author is Kasper, who post-mortem, these are different brains, established and reconfirmed the ideas by Konstantin von Economo that there are very different histological areas in the area of the TPJ. Okay, this is to say that we are already at the core of the problem. The TPJ is apparently not homogeneous, even from an anatomical point of view. Even if we establish the borders of the TPJ, when you get to the TPJ in detail, the TPJ is not homogeneous. If you look at the relationship with the white matter tracks that are below the TPJ, that connect the TPJ to the other areas, as you can see, there are several tracks. These are, for example, these are the different branch of the superior longitudinal fasciculus. This is the arcuate fasciculus. This is the inferior front occipital frontal fasciculus, and this is the inferior longitudinal fasciculus. So as you can see, the TPJ has a widespread anatomical connection with other areas with several branches and white matter pathways that connect the posterior parietal lobe to the frontal lobe, to the frontal area. This is also confirmed by connectivity studies. When you study the connectivity of these different areas at rest, you find that different sectors of the TPJ, these different sectors, have slightly different connection, functional connection in terms of brain connectivity. So also in this case, you see that, to recapitulate, the TPJ is now, the first difficulty was defining the borders of the TPJ. If you look even to recent papers, different authors established different borders, different for the TPJ. So the TPJ literally in the recent years changed this shape from other to other. Now there is a quite good consensus that establishes the borders of the TPJ as the ones I've defined before. And but once you get into the TPJ, even if you have established the borders, then you'll find a quite heterogeneous area in terms of histology, connectivity, functional connectivity, and anatomical connectivity. Okay, so this is the basic problem of the TPJ, and so the main problem of the TPJ is these are the anatomical premises. When you look at the enormous amount of works that have documented the involvement of the TPJ, both through the study of brain damaged patients or through the fMRI studies in healthy humans, you will find that the TPJ is involved in almost everything. So you find activation of the TPJ in memory task, in attentional task, in language task, in number processing, task of body ownership, self-agency of theory of mind. So it is apparently really I wouldn't say a mess, but a quite interesting and complicated problem. So it is involved in almost everything. It seems to do everything with TPJ. This is the basic problem up to now. And so this problem on the function of the TPJ has been recently, maybe thanks also to the description of different subsectors of the TPJ, more formally and precisely redefined in the following terms. The point is, do the different subsectors of the TPJ support different cognitive operations? So we have seen that the TPJ is involved in many cognitive tasks, but the TPJ also is made of different subsectors. So one hypothesis is that each one of these subsectors does something different as a function of the cognitive task that it serves, or the cognitive function that it subserves. But there is also another possibility, and the fact that the TPJ provides the same basic low-level cognitive function of cognitive operation, and that this operation is used in different ways by all these different subtexts and all these different functions. So in this second hypothesis, that the TPJ has a unique or a basic function, okay, that then is conveyed through different subsectors to different cognitive operation, okay? So this was the state of the art, and it is still is. I think we have provided, as I have the chance to show you, a quite satisfying explanation or hypothesis, if you want, on the function of the TPJ, but this has still to be considered, like has to be done in science, is still an open problem. Okay, so what I'm going to tell now, before going to explaining and describing our hypothesis on the function of the TPJ, is tell you how we reach this hypothesis. Okay, which are our empirical how the empirical findings we had in our lab with the years, because this has been a long-term, to say, consideration of the empirical findings before reaching the formal theory that we have advanced in review, how we reached this general idea on the TPJ. Of course, everything started for us, me at least, about 10 years ago with the study of attention, or spatial attention. Okay. I think everybody of you knows the Posner task, and which is using attention to study the shift of attention. In the task you have a fixation point, 2 little boxes, and in this version of the task that is used to test voluntary endogenous control of attention, you have an arrow, okay, that points to one of the 2 boxes in each trial. Okay, in this case, I won't tell you why we use a double arrow. This is for an experiment, is a sort of important experimental control in the study of endogenous attention. It is to avoid exogenous component. But let's say that you are a participant and you're told you pay attention to the box pointed by the yellow arrow. Okay, so you disregard the blue. In this case, the arrow point the right box. Okay, so you have for 1.5 seconds, while fixating, you pay attention to the box. Then there is a target that appears in the box, and your task is to push a button as fast as possible as soon as the target appears. When you know very well, I'm recapitulating things that I'm sure many of you know, that when the target appears in the box pointed by the arrow, the target is valid and you are extremely fast in responding. When the target appears in the box opposite to the one pointed by your arrow, you are slow. And then there is a neutral condition, a baseline condition. You see, these are double arrows. There is no arrow that has a single color, so you cannot orient your attention during the presentation of the arrow. And in this case, you have intermediate reaction time between the valid situation and the invalid situation. So you are fastest with the valid, intermediate with the neutral, slowest with the invalid. The difference between the reaction time between valid and neutral is called benefit. You have a benefit in orienting of attention. The distance between invalid and neutral is called cost. So one of the problems with this task is that in the majority part of cases, the 80% of trial is valid and that only the 20% of trial is invalid. So when you look at the difference between valid and invalid trials, what we are looking are 2 things contributing to reorienting of attention to the invalid target. One is the fact that the invalid targets appear in another place in space, so you have to reorient attention. But there is another important fact which is also important. These invalid targets are rare, are infrequent. So there is the appearance of invalid targets also triggers a novelty reaction. So this study was done to separate the spatial component, the reorienting, from the novelty component of reorienting. And to do this, we simply did this is the study, it's an old study. The same subjects performed the task in 2 conditions. In one condition, 80% of trials were valid and there were 20% infrequent invalid targets. In the other case, 50% of trials were valid and 50 were invalid. So in the second case, you have reorienting, but you have no novelty because invalid trials are as frequent as valid ones. Okay, this is the general design. And behaviorally, what we found is that when there is no novelty, there is a suppression of cost of reorienting and maintenance of benefits, but this is tangential to our argument, so I'm going to skip this. And the basic point is the fMRI activation we found. Okay, when we compared invalid trials to valid trials, so unexpected trials, something that introducing the argument of something that mismatches your expectancies, okay, compared to something that matches your expectancy. Because when a trial invalid, you were expecting the target on the right, the target piece on the left. There is a mismatch between what you expect and what you get from the environment. When it's valid, you pay attention to the right and the target piece on the right, you have a match. So your expectations are matched by the sensory environment. So when you compare invalid to valid targets, you find a classical finding that is the right hemisphere dominance in this case with the right TPJ, and this is important for argument, and these frontal areas, okay? But we did something different from other studies because we also used the comparison between invalid and neutral and valid and neutral, which have been rarely, if never, considered in fMRI studies. Once you do this, you find something really exciting, because when you compare invalid trials, so something you don't expect, a mismatch, to a condition where you had no expectation because you are unable to orient attention, you find a bilateral activation of TPJ. So the TPJ is active both in the right and in the left hemisphere. So in the left, also in the left hemisphere, there is a module that triggers and signals and codes the mismatch between what you expect and what you get from the environment, okay? And also important, when you compare valid, so what is expected, when your expectation is matched by the target, with a neutral condition, the condition where you have no expectation, there is a selective activation of the left TPJ. Okay? And we shall be back on this argument later on. And this activation is even more pronounced when valid trials are frequent. That is even more enhanced when the majority of trial is valid in 80% of cases. But anyway, the left TPJ codes also matches. So the summary of the result is the following. The right TPJ is mainly or exclusively activated by mismatches between what you expect and what you get from the environment. The left TPJ is activated both by mismatches, okay, and matches, okay. So the next problem is establishing, is this double response of the left TPJ to what you expect and what you were not expecting, mismatches, and what you expect matches, done by different population of neurons. So the answer is yes, because then we submitted this data we made another experiment and used multivariate approach to establish whether the pattern of an activation within the TPJ are the same or not for match, and within the left TPJ are the same or not for matches and mismatches, or are two different populations. And the outcome was that in the left TPJ here, you have two different populations that code for, one codes for match and one code for mismatch, okay? So this is the basic ground, okay? Why I'm talking about the role of TPJ in attention and attributing this activation to match or mismatch? Because the TPJ, this is another important notion, is fundamental to trigger a late response in the brain, ERP response in the brain, the P300, okay? And the P300 is particularly enhanced when something that you do not expect happens in the world, okay? And less enhanced with something that expect happens in the world. So it is well established through ERP studies and other kind of studies that the TPJ acts at a late moment of processing, in this case of the event, the target, okay? So the TPJ simply tells to the brain, the right TPJ discharge, let's say 300 milliseconds after an invalid target is appeared, and simply gives the signal to the brain to say, listen, what happened? The target did not appear where you were expecting. So it's a mismatching. okay? The left TPJ does both, okay? Has a population who codes the match, tells to the brain, okay, the target has appeared where you were expecting, and another population that in the case of spatial attention is whose action in the case of spatial attention is only limited to the right side of space, that tells, listen, the target did not appear where you were expecting. What is this for? This is the basic mechanism that the PJ, the basic info that the PJ gives to the brain so that the brain can build up a statistical representation of the coincidence between the cues and the target. That is, so that the brain can establish how much and how well cues, the direction of cues predicts the position of target. So this is the predicting role. This is the role played by the TPJ in what is, let's call it the predictive brain. So this is our two basic operations. Mismatch between what you expect and what happens. Match between what you expect and what happens in outside. So these are literally the basic, the foundation of our interpretation of the TPJ. So what actually happened was this. We had this data, then I kept on working on spatial attention. But at the same time, I was reading papers and papers and papers on the TPJ, and little by little, I was aware that this interpretation of match and mismatch could have explained almost all the activation you find in domains that are different from attention. So what I'm going to do now is literally taking the PDF of my review and scrolling little by little, because it's a long list and it would be impossible to summarize in detail in 45 minutes, and show you how we recollected the coincidence between this basic role we found in spatial attention and the same role that we found that the same role, match-mismatch role, can explain the involvement of the TPJ in disparate cognitive domains. Okay, so this is the premise, the conclusion of our initial work and the premise of the review. We use this data as an hypothesis to explain the activation you find in the TPJ by re-examining data, a large amount of data from the literature. And so what our idea is the following, to summarize. The TPJ support the brain representation of statistical contingencies among sensory, motor, and cognitive events, okay? The TPJ signals or gives a signal of matches and mismatches between expected and actual events, okay? The left TPJ codes both matches and mismatches between what you expect and what you get from the environment. The right TPJ is mainly, if not exclusive, involved in coding mismatches between what you expect and what happens in the environment, okay? Is it, I will stop now in the sense that are these premises clear? Is there any question? Because I'm ready to answer in case something is unclear.
If someone from the audience has a question, now is the time to write it in the Q&A, or if
or talk. You just talk. I prefer reading the chat sometimes. Please feel free to make questions before I go ahead.
From my side, there is no question. If someone would like to ask something.
Okay, can I go ahead? Okay, perfect.
1 second, there is a question. Sorry.
Yeah, okay. Who makes a question?
Luana? Oh, okay. So I will read it out. Can TPJ functions such as coding fits and mismatches be applied to understanding creativity and generating new ideas? How does this mechanism works? I don't know if you want to address.
The answer is yes. Is yes. This is a longer history, and the point is that the TPJ also, I'm talking about the role that the TPJ has when detecting the event. But the TPJ has also, and we go there, a role when you are expecting the event, which is a filter. It means like that. Imagine the cue is presented in our task. Let me go back. It's much easier. Okay. The cue, what I've been talking about now is the target, is the response of the brain to the target. Okay. But you also find the TPJ activation during the cueing period. Okay. When the cue are highly predictive, so when your brain has learned that if you pay attention to the right, there is a high probability that the target will appear on the right. Okay. In the right TPJ during the cue period, I'm not talking about the target period, I'm talking about the cue period, the right TPJ gets deactivated. Why? It's less active than when the cue are not predictive. And so the cue tells you, pay attention to the right, but you have learned that sometimes the cue is misleading, because when it says right, is also frequent that the target appears on the left. In this case, the TPJ is more active. Why this deactivation when you are sure, when the cue are predictive and you are quite sure that the target will appear on the side indicated by the cue? What is happening in this cue period, and you have a deactivation, is that all the representation of space which are different from the cued point are slowed down, okay, are shut off. There is a spontaneous activity in the brain. All the representational space are acting at the same time. When the cue is really predictive, you don't need to get activated the point of space that are different from the one that has been cued by the ARA, okay? So if you look at the general activation of the ARA, the activation goes down because a few neurons or few representational space remain active during the cue period, okay? When the cue is not predictive, and so you will probably need to reorient your attention, the brain learns this, and the TPJ maintains active also the representation of the points in space that are not indicated by the cue, because there is a quite good probability that the target will appear in those uncued points, okay? So this means that the brain activates everything, okay? It's less filtering out. What has to do this with creativity in case, okay? Creativity, and this has been demonstrated by several studies, takes in when you stop, or where you are no more slave, or when you suspend, when you stop your expectation, okay? Mind wandering means that you are not selecting one particular idea, so you are considering everything. So in this case, the TPJ would probably be shut off, okay? You're not selecting one particular idea because you are suspending, you're stopping your expectation to consider other possibilities. The same thing like in attention. So the role of the TPJ in creativity is probably this. The TPJ, when you are creative, when you are mind wandering, when you are finding new solution, is stopping expectation. So there is no particular thing to expect. So it has a different rate of action, different rate of functioning from when you are linked to a precise expectation. So you are not creative when you are fixed on a belief. So you think that something will happen and nothing else will happen, okay? In this case, it's the same thing as when you are filtering out all the uncued position in space when the cue is really reliable, because you know that the cue the target will appear with high certainty on the point indicated by the cue. So this is one of the link with creativity of the TPJ. But I think if we have time, because time is short, we will have the chance to talk this about in the context of language. So now I would like to go on, otherwise I won't have time to finish my talk.
Yes, please.
Okay.
Yeah. Okay,
so I'll skip this and I'll take literally, I'll show you you are still on the complete sharing of the screen.
Now we see your PDF.
Yes, it's fine, but you don't see anything else. Is what I'm. So this is the PDF. No, so let's go on through the different functions that we might consider through the match-mismatch function. So I'm doing this because there is a long list of things to be considered. Okay. Let me go to the theory of mind. Okay, TPJ and theory of mind. No, theory of mind is getting into the idea of somebody else. No, the typical task to study the fact that you can consider the idea of somebody else, the belief of somebody else, and this belief does not match with your belief. Is instantiated by the false belief task. I don't know whether any of you know this task, but there is a very, very more simple way of talking about this task. It's the Little Red Riding Hood story. Everybody know the tales. Okay. When a Little Red Hood arrives at the house of the grandmother, of the granny, and the wolf is inside the bed at the place of the granny. Little Red Riding Hood believes that the wolf is the granny, while you know that in the bed there is not the granny, it's the wolf. So there is a discrepancy, a mismatch between your belief and the belief of Little Riding Hood. Okay. And this is exactly why the TPJ is active. The right TPJ is active during false belief tasks and during the theory of mind, because you are aware of a mismatch between your belief and the belief of the character of the story in this case. Okay. The interesting thing is that in false belief task, you have an encoding phase when you hear the story. Okay. And then in task they make you a question about the story or what the reader writing will believe or what you believe. Okay, these are two parts of the task. And the interesting thing, in the second part, you have a left TPJ activation. Why? This is very easy to explain because many authors like Perner, which is an Austrian author who studied the law theory of mind, said that there are two things that are crucial in theory of mind. The first is detecting a mismatch between your belief and the belief of the character. The second is thinking that both beliefs coincide on the same reference. They are both belief on the same, on the same. In this case, is the wolf or the granny. Okay. So you must perceive both a mismatch between your belief and the belief of the character, and perceive a match on the fact that both beliefs are coincident, as Perner say, on the same referent in the story. So you have in this kind of task, you have both a right TPJ initial activation and then also left TPJ activation. So, and this is why this is argument. We are argumented that this basic function can also explain the TPJ activation in theory of mind task. So this is for theory of mind. Okay. This is another interesting thing. I will limit this consideration, given the limited time I have, to agency. Okay. The fact when you make a movement, you are conscious, you get conscious that you were the actor, the active actor of this movement, because you see a coincidence between what you have programmed, a match between what you have programmed and what you get. Okay. All the authors in this area, I'm talking about authors like Brass, Marcel Brass, or Chris Frith, have proposed that the distinction between something that you have decided to do, agency, or a movement that you didn't decide to do. The simplest example is that somebody moves your arm. You know that it's not you that you move your arm, because there is no coincidence, there is a mismatch between what you see and the program you made in the brain, okay? Another example is, in this case, you are able to distinguish if you were the agent or not of your action. So these authors say the distinction is based on the appreciation of mismatch between what you have programmed and the feedback you get from visual feedback. There is a very nice experiment by Brass showing that you have two fingers, use two fingers, and you see the same two fingers on the screen, corresponding fingers on the screen. When you have, let's say, a small number, you push left. When you have a high number, you push right, okay? And when you push, at the same time, you see on the screen, what are you pushing? Sometimes they manipulated what you see in the screen so that in the screen, the response is not the right one. So you have to inhibit imitation, make a distinction between other movement and your movement, and make the other movement. When you do this, that is, when you detect a mismatch between the other one, and when there is no congruency between what you program and the feedback you have on the screen, there is right TPG activation. You get the same right TPG activation when, for example, the corresponding movement that you see on the screen does not mismatch the movement you have made, but is delayed in time. So you make the movement, you see the other movement, let's say 1 second later. So there is still a mismatch, it's a temporal mismatch. So also in this case, the TPJ is active, the right TPJ is active, and all these authors says that this is because there is a mismatch between what you have programmed to do and what you see. So your agency, you feel that you are agent of what you do when there is a match between what you do and what you see, or what you have planned to do and what you see, and you make a distinction between what you do and what the other do when there is a mismatch, okay? Oh, episodic memory. Episodic memory is another nice thing. You get a left TPG activation when you are sure, especially the more you're sure that you have correctly recollected an item or rejected an item you have memorized before, the more the left TPG is active. And in this case, it's a perfect match. The more you're sure of the match between what you have recollected and what you have seen, the more the left TPG is active. Number processing, for example, the left TPG is extremely active when you see for mathematical facts, that is to overlearn operation. You know very well that 8 plus 4 is 12, and because you have overlearned this, and there is a perfect match, a constant match between what you have learned and what you see, and in this case there is a left TPJ activation. Language. This brings us to another argument. Language is classically a routine behavior. Routine behavior means that what you are saying or what you are going to hear is expected. What does it mean? If you, for example, hear somebody saying, and at the moment he says something wrong that does not match, you have a right TPG activation. Otherwise, you have constantly left TPG activation, because language is an overlearned routine. Okay, it's an overlearned routine, and nothing strange happens while you are talking or hearing somebody, except, for example, in case of humor or in case of metaphors. When you hear a metaphor like "he has a heart of stone," okay, this is not coincident with your semantic knowledge about the heart. Your heart is made of flesh, it's not made of stone, okay? And the metaphor is and in this case, you have a right TPJ activation, because there is a mismatch between what you hear and your semantic knowledge. Once you understand the metaphor, because you pose the metaphors between a conventional framework. Another kind of thing, let's say, I have an example here. If you heard the baby flies, this is totally incongruent. Baby do not fly. Okay, so in this case, you have a right TPJ activation. But if you are reading Peter Pan, okay, and you read the baby fly, you know that Peter Pan flies. You know, so in this case, there is a left TPJ activation because it is coincident with your knowledge, in this case, semantic literal knowledge of the story. Okay. So these are many, many examples. I won't go through that, except that I really want to talk about two things, and then I will stop and give room to questions. The first thing I want to talk is about the optimistic bias. You know what is the optimistic bias? Does anybody know what is the optimistic bias? If somebody asks you, what is your chance of getting healed by cancer? And you say 20%, okay. And the other replies, no, given your age, your health status, you have only 5% of chance of having cancer. And 1 week later, you are asked again, how much is the chance for you to have cancer? You say 7%. So you say 20 in the beginning and 7 in the end, after you had a bad news, a good news. For you is a good news because you believe it was 20 and it's 5. So you change, okay, you change once you have an optimist, a very good feedback, you change a lot your opinion in the optimistic direction because you believe it 20, they say 5, and you say 7. Okay, if you say again other condition, I have 20%, they say no, you have 50%. Okay. Bad news, and 1 week later they ask you how is your possibility to get cancer. If you had said 20% and they say 50%, you say 25. So you change very little. Okay. When you have bad news, you change little your opinion. When you have good news, you change a lot your opinion in the good direction. Okay. So this is the optimistic bias. Now there is the literature on optimistic bias says, for example, persons who have a huge optimistic bias, okay, are insensitive to bad news and have low right TPJ activation. Why? Well, bad news mismatch what you implicitly expect. Good news in this case. Why good news implicitly match what you expect. And there is a lot of evidence that the optimistic bias corresponds to fluctuates as a function of stimulation of the right or left TPJ in this direction. So you can enhance the mismatch. If the TPJ is overdriven, you get pessimistics because you are more sensitive to bad news. If the left TPJ is driven, you get more optimistic because you are more driven toward matches between what you implicitly expect, that is good news, and what is going on. I will finish here just by saying that the same applies to depression. And there is a long-standing issue in neurological literature on the fact that very often right hemisphere damage entails euphoristic, maniacal, hyper-optimistic attitude. Now, in the context of our hypothesis, this is really explainable. When you have a right brain damage, the mismatch module is off. So only for you, everything matches, okay? Vice versa, depressed people have overactivation of the left hemisphere, or there is EEG evidence, other kind of things, and this is explainable with an overdriven, or people with left brain damage get more easily depressed or have catastrophic reaction. Why? Because the match module has gone, nothing fits what you expect, and the mismatch module on the right hemisphere remains alone. So everything happens, mismatch your expectancies, and so you get depressed. So these are, I'll stop here. You can find a much more detailed. If you read this paper, I'm happy to send it, but I promise you, it's a long review. You will find a lot of details, a lot of information. So I'll just give you room to questions. This is a picture that summarizes a bit our ideas on the role of the left right TPJ and the role they play. And so the take-home message is left TPJ and right TPJ play this basic role of signaling to the brain matches or mismatches between what you expect and what happens. And this very basic operation, which is served to the predictive brain, can explain the involvement of the TPJ in several and different cognitive domains. And I'll stop here for a question. Hope everything was clear, because the review is quite complicated, but I invite you, I'll be happy if somebody of you goes through a detailed reading of the review, because it's really detailed, a lot of information. So to have detailed replies. On the journal, we already had some replies from colleagues that are published, and we publish an answer to their replies. So you can, if you go to the journal, you can also study in depth the debate that has been running on between our colleagues and ourselves, through their objection and to our general answer to their objection. So thank you again for inviting me. Now I'm happy to give answer to anybody.
Thank you very much for this wonderful and very insightful talk. We have a couple of minutes for questions, and I would start with the ones that we already have in the Q&A. So the first one is from Adrian, and it says, how do you filter out errors when you do the statistics? Is it possible on a living system, considering the fact that you don't have a reference for the model? Unlike a sensor where you know the minimum and maximum values, when talking about the brain, the limits are fuzzy, or am I wrong?
Could you repeat, please? This is a very interesting and complex question. A bit slower.
Sure. Sorry.
No, don't worry. No, go ahead.
How do you filter out errors when you do the statistics? Is it possible on a living system, considering that you do not have a reference for the model? Unlike a sensor where you know the minimum or maximum values, when we're talking about the brain, the limits are fuzzy.
Okay, the answer is quite straight. When I was talking about the TPJ, the focus of my talk has been mainly on the response of TPJ to target events. But I've been talking also, I made a short note, what happens during the queuing period. So when you are expecting events, what the TPJ does in this case ahead of the events is creating a model of the event. In fact, a complete version of our theory is that there are match or mismatch to template responses. So the model is built up based on prior previous beliefs. Is the DPJ builds a dynamic model which is modulated by the statistics of the environments ahead of what happens in the world. And so in the TPJ during the Q period, you have a model of what's going on by filtering out. When you filter out a position instead of another one, you are building up an expectancy, a template of what you expect. And then once you get the event, this event is compared to the template, which is already prepared in TPJ. So the TPJ has these two roles: a preparing role ahead of the appearance of the event, which means building up a template of what will happen soon, and a comparison between the model, the template, and what happens. And this mechanism is dynamic. So you update the model in the case of attention, for example, on the filter, the fact that you filter more or less out the uncued position as a function of what happens trial by trial. I hope this was clear.
Yes, thank you very much. Then another question is regarding neuroaesthetics and aesthetical judgments. Can this function of the TPJ explain the individual differences in attentional processes that activate subjective salience and attentional processing of the aesthetical object, arising the hedonic feelings of the aesthetical experience?
Well, we have a paragraph in the review where we show that, where we argue that since there are data that supports this, the TPJ developed by signaling matching or mismatching with what you are expecting, allow the development of hedonism. Hedonism is something that is not linked to immediate rewards. Okay, so the TPJ signals the mismatch between what you expect and what happens, independently of the fact that what happens is giving you a reward or not, independently of what is happening before is frequent or infrequent, etc., etc. So the fact that this system, which has developed late ontogenetically and develops late, who developed late in phylogenesis and develops late in ontogenesis, okay, get in some way detached from immediate reward, so that you can develop a system that gives you pleasure for the fact of being right in something. Okay, so in the case of aesthetic appreciation, this is a good question, a very good question, and I think that the problem could be framed along this interpretation. But in this case, I will just take the suggestion to check the literature and see, and I believe it's like this, whether this response of the TPJ can fit or explain the aesthetical experience. My belief is yes, but I have to check. Honestly, this is a very, very good suggestion, because notwithstanding the fact that we have been trying to check all the literature, there are some things that have been left out. For example, if I have a minute, there's a very nice study by is a friend working in Munich on rhetorics. Okay, on rhetorics. Then on advising, and when there is no correspondence between what you get from advice and what you expect as advice, you have a right TPJ activation. So even in rhetorics, somebody who says something which is out of the general belief in a group is going to activate the right TPJ in everybody. This is the point, because it mismatches the belief of everybody. Yes.
Yeah.
So as you see, you can see this, just to say that this very basic mechanism is involved in a very, very disparate cognitive operations, a pervasive mechanism.
Yes. Thank you again very much. I think we have time for one final really quick question, and afterwards, unfortunately, we will have to move on. So I would read another one from the chat. Catalina is asking, are there potential cultural or societal influences on the development and expression of positive biases in the TPJ, and what are the implications for cross-cultural psychology?
Oh. Well, of course, the response is what this has to do with belief, with the system of belief, which is linked to culture. So, is in a way, I already, not to be too short, but what I've said before. Imagine you have 6 persons in a room, 5 belonging to one culture and another one belonging to another culture. At the moment, for a social convention, the only one belonging to different cultures makes something different from the others. Well, the others, the right TPJ is going to be activated there. So since it has a late ontological development, of course, the TPJ is susceptible to modification of its activity, or frames its activity based on the inputs you receive, the feedback you receive during education.
All right. Thank you very much again, Professor Doricchi, for.
It was a pleasure to be here. I hope everything was clear. I'll be happy, but you can find my address if anybody has to ask any question or have a desire to have a copy, a PDF copy of the manuscript, and he is unable to download, even if it's free, is open. You can write to me. Okay.
Yes, we will try to collect the questions from the chat and find another format in which we can answer them.
Perfect.
Ask our audience to understand this.
Thanks for your invitation. It was my pleasure.
Thank you very much.
Have a nice day. Bye.
Have a nice day. Goodbye.
Bye.